Hello friends, welcome back to our YouTube channel Doctor's Corner. Today we are going to discuss one of the very important topic in the physiology of central nervous system, that is the very basic topic synapse. Okay, so we'll cover this topic synapse in two sections, that is part 1 and part 2. In today's section of part 1, we'll discuss about the definition of synapse, what are the different types of this synapse, what is the structure of a chemical synapse and how the transmission takes place in a chemical synapse, and finally what are the types of inhibition in synapse. Okay, and in the next lecture, it is the part 2 of the synapse, we will discuss about the properties of the synapses. Okay, from exam point of view, for the theory exam of first year MBBS physiology, many a times this question has been asked for 10 marks question, describe the synaptic transmission or describe the properties of the synapses, or even for a short notes describe the EPSP, that is the excitatory postsynaptic inhibition, or IPSP, inhibitory postsynaptic inhibition, or sometimes the Renshaw cell inhibition, 3 marks question, whatever. We'll discuss in detail about this thing in the 2 sessions. So please subscribe our channel Doctor's Corner, and we are going to distribute the notes also in our Telegram channel. Just after this session of after the second video, release of the second video, we'll just share the handwritten notes. Okay, so please give your valuable feedback and share the link to your friends. Okay, coming back to the synapse, what is a synapse actually? What do you mean by synapse? So it is nothing but in the simple one line definition, it is a functional junction between the two neurons. Okay, more specifically it's a functional junction. Okay, or you can just say it's a junction. So you know our central nervous system is made up of so many neurons, right? Or the nervous system, so millions of neurons are there. So you know a typical neuron has the neuronal cell bodies, processes, that is the short processor dendrites, and one long process, that is the axon. Okay, and it is covered by the myelin sheet, and at the peripheral end there is a smallest bulging that is known as synaptic knobs. Okay, this is one neuron. So this one neuron will communicate with one more neuron, okay, one more similar neuron. It is the one cell of the nervous system will communicate with the another cell of the nervous system, and the junction between the two cells of the nervous system, that is the junction between the two neurons, is known as synapse. That is the definition. And it's not the anatomical junction, it is a functional junction. Okay, there is actually no anatomical continuity between that, mainly it is a functional junction. Okay, that is the synapse. Now what are the different types of the synapse? Okay, first what we have studied now is, first thing is definition. We have studied what is the synapse. So functional junction between the two neurons is a synapse. Now secondly, we will study what are the various types of the synapses. So classification of the synapses can be classified broadly into 2 groups. One is anatomical types and another one is physiological types. Okay, anatomical and physiological classification, or anatomical types and physiological types. Okay, so anatomical types are of the synapses are mainly, there are many, but will main types what we study is exodendritic synapse. Secondly, what is exodendritic synapse? That is axon of one neuron will communicate with the dendrite of the another. That is the communication or the junction between these two, axon of one neuron and the dendrite of the another neuron, it is known as exodendritic. Another one is exosomatic. Mainly the exodendritic is the most commonest one what we see. Then we have the exosomatic synapse. Okay, so that is axon of one will communicate the nerve cell body, that is the soma. Okay, that is the communication between that, it is termed as exosomatic. Then we have the exoaxonic type, that is the axon of one neuron will communicate with the axon of the another neuron, that is known as exoaxonic type, which is typically rare. Okay, so apart from this, even we have the dendrosomatic, dendroaxonic and all, but commonly we see this type of the classification with this type of neuron. Most common is the exodendritic type. Okay, then physiological classification, again it's very important. Mainly it is of 2 types again. One is chemical synapse, another one is electrical synapse. So what is chemical synapse and what is electrical synapse? So the chemical synapse means the message from one neuron to the another neuron is transmitted by the release of one chemical substance that is known as neurotransmitter. That is, that type of synapse, it is known as chemical synapse. So in our central nervous system, most of the synapses are chemical synapse. There is one more type, electrical synapse. Instead of releasing a chemical substance at the nerve endings, there is direct communication by the gap junction through which the ions can be transported, and the message from one neuron is transported to the another neuron. That type of classification is known as electrical synapse. So we see this type of synapses in the olfactory bulb or in the retina, okay, and sometimes even we see both together, that is known as conjoint, okay, both chemical and electrical synapse at a similar junction, okay, at a similar point. So these are the different types of the synapse, okay. So we have studied about definition and types of the synapse, okay. Now we'll discuss about the chemical synapse in detail, okay. So there are differences between electrical and chemical synapses, so we'll share the details in the notes, okay. Time won't permit me to tell in detail about that because the video length will be too much, so I will be just concentrating on the important aspects about the chemical synaptic transmission. So just to highlight what the difference mainly between electrical and chemical. Electrical synapse is more the transmission is, that is the conduction speed is more fast, and the chemical synapse, the conduction speed is relatively less as compared to the electrical synapse, okay. And this chemical synapse, there are chances of fatigue, and they are exposed when they are exposed to pH and all, so they will get the transmission is delayed, okay. So now definition and types is over. Then the third thing what we'll study is the structure of a chemical synapse, okay. Structure of a chemical synapse and how the transmission takes place, okay. So first thing is I'll just draw a presynaptic neuron, okay. Presynaptic neuron. Another one is postsynaptic neuron. So the junction between these two is known as synapse. So what is this presynaptic neuron? What is this postsynaptic neuron? Just now I have said you, the neuron from which the signal has to be transmitted, okay. So imagine this is a neuron A, and another one, this is a neuron B, okay. Neuron B, it has in roughly I'm drawing this. So the signal from this neuron A to the neuron B has to be transmitted, that is the junction, the terminal endings, that this is known as terminal knobs, synaptic knobs, okay, terminal knobs, and it is communicating with the dendrite of the next neuron, okay. So the axon is communicating with the dendrite of the next neuron, and this junction, if we enlarge and see, so that's the chemical synapse what I am showing a typical. So this is the terminal knob of a axon, okay, and this is the dendritic next process of the neuron B. So the space between these is known as synapse typically. Now this membrane it is known as presynaptic membrane, okay. At the junction, the neuronal cell membrane of the presynaptic neuron, it is known as presynaptic membrane, okay. Presynaptic membrane. And this membrane which is communicating with the other neuron, this membrane, it is known as postsynaptic membrane, okay. Presynaptic membrane or the postsynaptic membrane. And the space between this presynaptic and postsynaptic membrane, the space, it is known as synaptic cleft, okay. This is known as synaptic cleft. It is around 20 to 40 nanometer dimension, okay, synaptic cleft. The space between the presynaptic membrane and the postsynaptic membrane, it is 20 to 40 nanometer, okay. Then what else, if we see the ultrastructure, what else, what are the other things? So this synaptic cleft mainly consists of the ECF, that is the extracellular fluid, and some glycoproteins and all, and this is the presynaptic membrane. So in the presynaptic membrane, the inside is ICF, that is intracellular fluid, mainly the exoplasm and all. So what are the special things what we notice here is, here there are synaptic vesicles, terminal nerve endings. They are synaptic vesicles. Inside that, the chemical substance are present. That chemical substance are known as neurotransmitters. Okay, so there are again two types mainly seen. One is circular, which consists of the excitatory neurotransmitters, and flat, which mainly consist of the inhibitory neurotransmitters. Okay, excitatory neurotransmitters are glutamate, inhibitory are glycine and GABA, most commonly. Okay, so apart from this synaptic vesicles, okay, these are the synaptic vesicles which consist of neurotransmitters. There are other structures like mitochondria. Okay, neuronal, this one cellular organelle is like mitochondria, and one more very important structure here to be noted is microtubules. Okay, microtubules are also present in this presynaptic neurons. Okay, so these are the structures which are present, and this membrane is bit dense here. Okay, this will be dense zone here, and similarly this postsynaptic membrane, it is the exoplasm or the ICF is more dense here. Okay, in the at this junction, so dense zone. Now here in the postsynaptic membrane, what special things what you will see here is mainly the receptors. So the postsynaptic receptors. So mainly these receptors are again of two types. One is ion channels, ligand gated ion channels like sodium channels or chloride channels or potassium channels, or sometimes we see some receptors which are enzymatic in, that is whenever they are stimulated, so some enzymatic reaction takes place and sequence of events takes place that ultimately leads to the protein synthesis by the transcription and translation processes. Okay, and this whenever they are stimulated, these ion channels may get opened and there is a transport of sodium, chloride and potassium or other ions movements will be there across the cell membrane. Okay, so mainly this is the ECF is this synaptic cleft, and ICF is this one. Okay, so this is a typical structure what I have just highlighted, the very important aspects of the structure of a chemical synapse. Okay, now the fourth thing is how the synaptic transmission takes place in a chemical synapse. Okay, this can be a 10 marks question. Synaptic transmission, that is how the message from the one neuron is transmitted to the another neuron. Okay, so just to make this interesting, I'll just tell you one common example. For example, if a touch sensation or pain or any sensation, temperature sensation from outside, that is from the periphery, has to reach to our brain. Okay, for example, I'm closing my eyes and I'm touching or pinching my skin here, so I'm feeling that pain sensation or pinching sensation. Now this message, that is this pain sensation or even the just simple touch sensation, is carried to these touch receptors of the skin, which are carried through the neuron, and ultimately it will go to the spinal cord. Here it will synapse with one more neuron that will ultimately goes to the some pathways. So we will discuss about the different pathways, touch pathway and all, but timing remember. So how the signal goes to our brain, and simultaneously if I want to, that is the sensory signal, and the motor signal means suppose if I want to lift my hand, now I am lifting my left hand, right? So the again message from my motor cortex or basal ganglia or cerebellum, which will control the motor system. So that also from that message will go through the different tracks to the ultimately spinal cord and through this brachial plexus, ultimately to that concerned muscles and joints, and it will move accordingly. Okay, so this message transmission occurs, that is from one neuron or from one neuronal cell to another neuronal cell by means of the transmission, that is synaptic transmission. So synaptic transmission is very important to know, and from exam point of your first year MBBS physiology can be asked as a 10 marks question. So there are various steps how the exactly a message from one neuron can be transmitted to another neuron. Okay, so first step here is first the action potential, that is when this type A neuron, that is when the first neuron is excited, that is the presynaptic neuron is excited. So the action potential will causes the opening of the calcium channels here in this presynaptic neuron. Okay, so the calcium channels were open, and from the ECF calcium will enter into the ICF here inside, and due to this more amount of the calcium here, this calcium will causes the synaptic vesicles to come near, it will drag the synaptic vesicles to the presynaptic membrane here. Okay, now due to this dragging, and it will ultimately by the process of exocytosis, the synaptic vesicles come into the synaptic cleft, and finally they will burst and it will release the neurotransmitters inside the synaptic cleft. Again, neurotransmitter is of two types, excitatory and inhibitory. Okay, so the first step is release of this neurotransmitter into the synaptic cleft by the depolarization of the due to the opening of the calcium channels. Okay, so the neurotransmitter which was stored in the synaptic vesicles is released here. Now if the neurotransmitter is excitatory, then it will lead to some electrical changes in the postsynaptic neuron or the next neuron. That is, electrical changes, not the action potential, that will not transmit or conduct it. It is a local response, and that local response, it is called as EPSP, excitatory postsynaptic potential. Okay, so whenever there is an excitatory neurotransmitter, for example glutamate, which is the most common excitatory neurotransmitter in the central nervous system, so this excitatory neurotransmitter which is released at the synaptic cleft, it will bind to this either sodium or calcium channels, mainly the sodium channels, and it will open the sodium channels. So when it opens the sodium channel, so what will happen, sodium from the ECF will enter inside the ICF, that is the inside of this cell will become more positive, okay, due to the entry of this sodium. So for example, if the resting membrane potential is say minus 70 millivolt, okay, resting membrane potential is minus 70 millivolt of this postsynaptic neuron, that is the second neuron, okay, it is minus 70 millivolt. Okay, now if it is minus 70 millivolt, when, so it is minus 70 here, it is minus it will be minus 60, minus 50 and all, then minus 40, 0, here it is minus 80 millivolt. Okay, so if it is minus 70 millivolt at the resting condition, when the sodium enters inside, that is it will become more positive, it will become more positive. So the interior will become positive, and the value is around 8 millivolt deflection. Okay, from minus 70 it will become minus 62, that is plus 8 millivolt for about 4 millisecond. Again it will come back to the resting membrane potential. Just a small amount of change, electrical current is generated, that is known as EPSP. What is excitatory postsynaptic potential? So when an excitatory neurotransmitter like glutamate binds to the sodium or calcium channels in the postsynaptic membrane, and there is an entry of the sodium ions, it causes some local response or some local current. That local response or local current of smaller magnitude, it is known as EPSP or excitatory postsynaptic potential. Remember, it's a local response, not an action potential. It does not obey the all or none law and other properties of action potential. Okay, so it is a smaller current. Okay, so similarly we have one more term here, IPSP. So what is IPSP? So IPSP is known as inhibitory postsynaptic potential. From this terminologies only you can understand. Okay, sometimes can be a 5 marks or 3 marks question. What is EPSP or what is IPSP? Inhibitory. So when a inhibitory neurotransmitter like glycine or GABA means gamma aminobutyric acid. So this inhibitory neurotransmitters binds to the postsynaptic receptors like chloride and potassium. So when it binds to the chloride and potassium, what happens is chloride ions enter inside, that is the interior will become negative, and the potassium ions tends to move outside. Okay, then the interior will become again negative due to the removal of the positive charge, that is potassium. So it causes the hyperpolarization, that is if it is the resting membrane potential was minus 70, it will become after a lag of delay of minus 2 seconds, that is synaptic delay. We'll discuss about that in the properties of the synapse. So it will become minus 72 millivolt. Okay, and after that it will come back. So this is known as IPSP, that is inhibitory postsynaptic potential. Again it's a local potential, but it is a hyperpolarizing potential. And this one is EPSP, which is a magnitude of plus 8 millivolt. This is just IPSP is just around minus 2 millivolt. Okay, and it is due to the opening of the chloride or potassium channel. So mainly the interior will become negative here, it will come to minus 72, here it will become to minus 62, the interior becomes positive, then it is EPSP. Now how this local current will form the action potential? So the next step here is, first step is release of the neurotransmitter due to the opening of this calcium channels due to the action potential in the membrane in the postsynaptic neuron. Then this neurotransmitter which was released in the chemical synapse, it will either generate EPSP or IPSP or both. or both, okay, and that all this will be summed up. EPSP and IPSP will be summed up, and ultimate effect, either EPSP or IPSP, ultimately effect will be there, and there is a formation of action potential. So how this action potential is generated in EPSP is from the EPSP is by the property of summation. Okay, we'll discuss in detail about that property of summation when we discuss the properties in the next lecture. So timing, remember that summation of these smaller potentials, local potentials, takes place. So this summation is again of two types. One is temporal summation, another one is spatial summation. Temporal and spatial summation. What is temporal summation is when the smaller EPSP potential, there is repeated stimulation of this repeated neurotransmitter is released, glutamate, and it causes a repeated EPSP production. Then due to that repeated inhibition stimulation, this will get summed up. The plus 8 millivolt will get summed up, and they will cross the threshold, and action potential is generated in this neuron. So that is this, due to the summation, the action potential is generated. Okay, so temporal is repeated stimulation, and spatial summation means whenever the smaller EPSP, there are many smaller EPSPs are generated across these different synapses, then that are known as spatial summation. We'll discuss in detail about this. Okay, so this is how the chemical transmission takes place. First thing is release of the neurotransmitter by the opening of these calcium channels and the moving of these vesicles here. Then the neurotransmitter binds to this and formation of IPSP and EPSP, then finally the summation, which leads to the action potential generation, which goes to the next neuron. Okay, so this is the chemical transmission. Okay, in notes we are given in detail how exactly it is there. Now finally we'll study about the types of inhibition. Okay, very important. With this last 5 minutes I'll finish it off the today's lecture, then we'll discuss about the properties of synapse in the next video. So there are types of inhibition. Types of inhibition means in the synapse the impulse from one neuron is inhibited to the another neuron. Okay, so what are the different types of inhibition? So again, it can be a 5 marks or 10 marks question. It is asked how the synaptic transmission that is inhibited. So there are mainly 4 types of inhibition seen in the different types of the synapses. So the first one is postsynaptic inhibition or the direct inhibition. Postsynaptic inhibition or direct inhibition. Second is presynaptic inhibition or indirect inhibition. Third one is Renshaw cell inhibition or feedback inhibition. And finally the feed forward inhibition. Okay, feed forward inhibition. So just I will tell briefly about these types of inhibition. First is postsynaptic inhibition, all known as direct inhibition. What happens here is, so one neuron, that is this neuron 1 and neuron 2. This is neuron A, this is neuron B. So this neuron B is inhibited by this neuron A by the generation of this IPSP, that is inhibitory postsynaptic potential. That is inhibitory neurotransmitter is released from this neuron A, which will causes the inhibition of this neuronal B, that is hyperpolarization of this membrane, that is known as direct inhibition. Okay, by generation of IPSP. Then presynaptic inhibition. So the postsynaptic neuron, that is neuron B, is okay, but the excitatory neurotransmitter which are released from A is reduced, and the sensitivity of neuronal B will be okay, the receptors here will be okay, but from this neuronal A, the neuron will release less amount of the excitatory neurotransmitter, that is known as presynaptic inhibition. Why? Means there is one more neuron, for example say third neuron, neuron C, which will again synapse with the neuron A, and it will cause certain changes here in this local membrane, so that excitatory neurotransmitter is released less to the neuron B, so that the message is not transmitted here. That type of inhibition known as presynaptic inhibition. How exactly it will do? There are many ways. One is either they can block this calcium channels opening, okay, so that the release of the neurotransmitter is inhibited from that, or they will open the chloride channels, so that this will become negative inside and depolarization process does not causes the release of the neurotransmitter to the vesicles. Okay, or through the stimulation of the enzymatic stimulation of the G protein. Okay, there are various methods. That is the presynaptic. Then the feedback inhibition, also known as Renshaw cell inhibition. Renshaw cell inhibition. So in the Renshaw cell inhibition, how exactly the inhibition takes place is, so if a neuron A stimulates the neuron B, right? But again this neuron B will stimulate a neuron C, that is a interneuron, also known as a Renshaw cell. This interneuron, that is the Renshaw cell, what it will cause us, it will again inhibit the neuron B. So that type of inhibition, it is called as, by an interneuron, it is called as Renshaw cell inhibition, RENSHAW. Okay, Renshaw cell inhibition or feedback inhibition. That is, neuron A has stimulated the neuron B, but neuron B will stimulate a neuron C. That neuron C, which is an interneuron, will cause inhibition of neuronal B only. That is known as feedback, that is Renshaw cell. Then finally feed forward inhibition we see in the cerebellum. So we have given a good lecture about the cerebral circuits, so go through that video, but timing, remember, in cerebellum there are different types of neurons. So I'll just tell you one few types of neurons. One is granular cell. Okay, so granular cell will stimulate the two different neurons. Okay, so two different neurons here is, one is basket cell. Okay, one is basket cell, that is again a neuron. Another one is Purkinje cell, again it's a neuron, Purkinje cell. Now this granular cell will stimulate both this neuron, basket cell and the Purkinje cell. But what's the special here is, this basket cell in turn will inhibit the Purkinje cell. So ultimately, if you stimulate the granular cell, this Purkinje cell is also stimulated, but simultaneously due to the stimulation of the granular cells, Purkinje cell is inhibited too. So this type of inhibition is known as feed forward inhibition. How it is inhibiting the Purkinje cell is by stimulating is one more neuron, that is the basket cell, that will inhibit this Purkinje cell. Okay, so this type of inhibition known as feed forward inhibition. Okay. So that's about the first synapse, section 1. So just to summarize what we have studied today, we have studied about the definition of the synapse, types of the synapse, anatomical and physiological types, then the structure of a chemical synapse, then the most important is the synaptic transmission, and finally we have studied about the types of inhibition. In the next section, we'll study, that is, properties of synapses in the synapse 2. Okay, thank you very much.